Okay, so we're going to start your final review. So our first question is compare ATP and ADP for our photosynthesis chapter. And this is kind of like the first time we were talking about chemical energy. And so ADP and ATP are going to be adding and removing phosphate groups, so there's kind of a cycle to it. So if you have ATP, it's kind of like our main energy molecule. And if you remember correctly, ATP is adenosine, which is similar to one of our nucleic acids, and then triphosphate. So triphosphate just means 3. We have this sugar molecule right here, and then we have 3 phosphates, which is PO4, phosphate. So those are 3 little molecules, a sugar, and this is adenosine, so this is adenosine triphosphate. Okay, so when ATP is used, it is going to remove a phosphate, so a phosphate is removed. Okay, and when a phosphate is removed, we make ADP, which ADP is used for storing energy until it gains another phosphate, and it is adenosine diphosphate, di meaning 2, so it has 2 phosphates on it instead of the 3. This is still adenosine. And then you know, there's many different ways we can get phosphate added, but one of the common ones is that ATP synthase molecule that we find in the mitochondria and in the thylakoid membrane that generates hydrogen ions that attracts phosphate molecules that then puts in another phosphate ion for ADP to turn back into ATP. So these are molecules that transfer energy within our cells, within plant cells, and they are very important. Okay, so next one, where does most energy come from for life on Earth? Well, this is going to be the sun. Okay, so in order for anything to happen on Earth, anything to be living, we have to have sunlight, because the sunlight gives energy to our autotrophs, or to our plants, and these plants are then what everything else eats. So the herbivores eat these, and then our consumers, our tertiary consumers, eat our herbivores, and without the sun, none of this other would ever take place. Alright, so the next thing is asking us about photosynthesis. So photosynthesis is a really big process. Takes place in the thylakoid membrane. Hopefully you guys have a video of you explaining this, so that might be a really good way to review. So photosynthesis takes place in the thylakoid membrane inside of a chloroplast. So we have a chloroplast, and inside of the chloroplast we have these things called granum, and the granum actually is where this whole thing is the granum. It's a stack of the thylakoids, and inside of the thylakoid is the thylakoid membrane. So if we were going to blow this a little bit up here, the thylakoid membrane is going to be made up of little tiny phospholipids that are all in a circle that have little proteins inside of them. And in the thylakoid membrane, this is where the chloroplast absorbs the sunlight, and this is what we were just talking about. The sunlight goes in, water goes in. This is why we have to water our plants, right? We have H2O and sunlight coming in here, and during the light-dependent reaction, which we're going to talk a little bit more about, oxygen is produced, O2. And if you remember our photosynthesis equation, was that we use 6 H2O plus 6 CO2 yields 6 O2 plus 1 sugar, which was C6H12O6. And so oxygen is produced, and this is all within the light-dependent reaction. And then in the stroma, on the outside, in between areas of the thylakoid, is where the Calvin cycle takes place. And the Calvin cycle is going to be where we change the carbon dioxide that is going into the plant. This is from us breathing. Goes into this open space of the stroma, and the CO2, 6 CO2 to be precise, go through the Calvin cycle. And we produce our sugar, our C6H12O6. And so I kind of covered number 5. So here's our equation. So 6 water molecules plus 6 carbon dioxide produces 6 oxygen and 1 sugar. And then it says explain the transfer of energy in the light-dependent reaction. So to do that, we're going to have to get a little bit more detailed with our drawing here. So I already have a little thylakoid membrane started, so we'll keep with that one. Okay, so we have the thylakoid membrane, and in the thylakoid membrane we have some really important proteins. Our first one is called PS2, photosystem 2. And here we have our little chlorophyll molecules. Okay, and we have photosystem 1. And we kind of draw, and then we're going to have our ATP synthase, which kind of is this bigger molecule. I don't have the greatest phospholipids, but I think you get the point. So it's going to be in a circular pattern here. And then, of course, we would have phospholipids on the other side. And this is the thylakoid membrane. Okay, so what happens is the starting of all of it, sunlight hits our photosystem 2 right here, and it's absorbed. And we have hydrogen ions that are made from a water molecule being broken apart by the energy that this sunlight is producing. So we get an electron here that's high energy that breaks apart this water molecule, and we get hydrogen produced, and we have O2. And this O2 is going to be what is leaving the plant that in turn we're going to breathe. So these hydrogen are going to continue to build up here from the breakdown of water. We're going to get more O2 being produced and leaving the thylakoid. And this electron is going to travel over to our second energy, or our second protein here, our photosystem 2. And here we're going to create a little bit of energy with NAD. We're going to actually take some energy there, add an electron, make this an energy carrying molecule, and we're going to produce NADPH, which we're going to need in the Calvin cycle. And then when these hydrogen get built up high enough, they're going to start coming outward, and the hydrogen is actually going to attract phosphate. And if you remember, phosphate is what is important for ADP to turn into ATP. And so ADP latches onto this phosphate because phosphate is a -3 ion and hydrogen is a +1 ion. So it attracts them, and then that's why they're concentrated here, and we form ATP molecules. And that's what happens in our photosystem. So first, energy is absorbed from sunlight. So if we're going to do these steps, sunlight is absorbed. And then second thing, water molecule breaks apart. And that's creating the oxygen. And then the last one, hydrogen ions are transported. So hydrogen ions transported, and this produces ATP at the end. So those are the three basic steps of what's happening. Also, the NADPH. So two energy molecules, really, ADP and NADPH. So that is your light-dependent reaction. The next one is, how is carbon transferred for CO2 into a sugar during the Calvin cycle? And so this is the second cycle of photosynthesis that is really important. The Calvin cycle, remember, is again taking place in the stroma, so it's on the outside of the thylakoid membrane. And we're going to start out with a three carbon molecule, or excuse me, 6 carbon molecule. So we have a 5 carbon molecule that's going to start off everything. I was thinking of the Krebs cycle. These two kind of mirror each other, but they are very different. So we have CO2 that comes into the Calvin cycle, and this carbon is going to attach onto that, and this is O2 is going to be leaving. So we get a 6 carbon right off the bat. And this 6 carbon molecule is going to have it's going to be very unstable, and so there's going to be energy or electrons that are reconfigured, and when this happens, we are actually going to get some energy being used. So we're going to have some ATPs that we created in the light-dependent reaction turning into ADP, and our NADPH, that was high energy, being turned back into NADP, which this NADP will head back over to the electron transport chain to become turned back into high energy to continue this process in this cycle. And so when we come around here, we're going to pluck one of these carbons off, and we're going to regenerate this 5 carbon molecule. And it's going to go through a couple more reactions. ATP is going to be used again to reconfigure. And it's going to go back up here and grab another carbon dioxide. So remember, there's going to be 6 carbon dioxides. So this cycle is going to turn a total of 6 times, which is going to end up dropping off 6 of our carbons, which is going to build our sugar molecule. But every time one carbon is plucked off, and then one carbon from CO2 is put back on, and then it's taken off, and then it's put on, and then taken off. And this process continues until we have 1 sugar produced from 6 CO2, and that's just what our equation says. That we need 6 carbon dioxides to make 1 sugar. And so the Calvin cycle uses ATP and NADPH from the light-dependent reaction to produce the high-energy sugar that then us humans will eat and get energy from. Okay, the next one says, what affects the rate of photosynthesis? So there's a couple of things that can obviously affect the rate of how photosynthesis is taking place. So if we increase the amount of sunlight, increase the amount of water, increase the amount of oxygen that was available to the plant, photosynthesis could be done at a higher rate. Or if you wanted to slow it down, decreasing those things would have the opposite effect. Okay, so let's scroll down here. and start cellular respiration. Okay, so cellular respiration is going to be what happens in animals, so in animal cells. This is going to be taking place in the mitochondria. And the mitochondria has these little folds in it, and we call it the matrix here. So we have on the outside of the mitochondria, glycolysis takes place. And glycolysis is our first cycle. And then in the matrix, we have the Krebs cycle that takes place in here. And that will be the next one if oxygen is available. And then after the Krebs cycle, right here on the membrane, we have the electron transport chain, which is similar, but notice it's last in the cellular respiration, where in a plant it's first, because it's dependent upon light. But here it is not, so it's a little different, but reasonably similar. Now, I said glycolysis, what happens after that is Krebs cycle if there is oxygen. If there is no oxygen, then we call that an anaerobic environment. What happens is fermentation, and fermentation is the production of lactic acid. So I'm going to go through each of these with you to start off with, to maybe jog your membrane. Also, your memory. Anyway, fermentation can also take place to make alcohol, and fermentation can take place that way, which I will also be showing you. Okay, so the first thing we're going to talk about is glycolysis. So glycolysis, we're starting out with the sugar that we ate from the plant. So we start out with our C6H12O6, and this is called glucose. So we start out with glucose, and the first thing that's going to happen is this glucose is going to be broken down, and so it's either going to take energy or produce energy to do so. And so in the first part, we're going to use some energy from ATP, and we are going to, of course, produce ADP, but we're going to produce a very unstable molecule, 2 3-carbon chains, and they have kind of extra electrons on them. And these extra electrons are going to be plucked off, and so we're going to turn 4 ADP molecules, and we're going to use these electrons to make ATP, 4 ATP. And by the way, this is 2 ATP that turn into 2 ADP there. Okay, so with this, we end up making 2 pyruvates. So this is 1 pyruvate, and then we make another pyruvate. And this pyruvate is going to be what is taken to the Krebs cycle, and this is what is processed there if there's oxygen. So if it is an aerobic environment, we head to the Krebs cycle. If it is an anaerobic environment, then we will go into fermentation. So this would be O2 is present, this is no O2. And so anaerobic environment is going to be what the cell does, like when you're breathing really hard, so you're working out and you're burning all these carbohydrates that you've eaten, and that's great, and then you start breathing so hard you feel like you're going to collapse, and that's when we have no oxygen. We can't get enough oxygen into our bloodstream to power glycolysis. So what starts happening is these 2 pyruvate molecules are then transported into 2 lactic acid compounds. And so right here we actually produced 2 NADHs, 2 energy molecules. We've taken some of these. So we made NADH, 2 of them. And we're actually going to take this and we're going to use its energy. We're going to produce 2 NAD positives again, which is an energy carrier, and those electrons change pyruvate into lactic acid. So this is lactic acid. And lactic acid is going to stay stored in your muscles until you get it out of there, and it goes into the bloodstream, back into the liver, and after it goes into the liver, it turns back into glucose, and then it starts to cycle again. Okay, so to answer these, where does cellular respiration take place? This is going to be inside the cell, around the mitochondria. That's why the mitochondria used to be called the powerhouse of the cell. What are the three processes that make up cellular respiration? That would be glycolysis, Krebs cycle, electron transport chain, and then the process that occurs when there is an anaerobic environment, that is going to be fermentation, what we just talked about. So the products of cellular respiration then, so our equation is 6O2 plus 6 water produce, nope, sorry, just kidding. 6O2 plus C6H12O6. So this is the sugar that we eat. The oxygen that we breathe is going to produce 6O2, and that is in the Krebs cycle. That's why the Krebs cycle spins 6 times. And 6H2O. That is produced in the electron transport chain. Alright, so next thing on our list is cellular division, cell growth and division. That's the process of mitosis. And mitosis is really important for us to understand because this is how cancer is reproduced and becomes such a problem. And so let's get started on that. So what are the stages of the cell cycle? Okay, so if we draw out a circle here, stages of the cell cycle, we're going to start out with one cell, okay, and the whole entire point of mitosis is to make an identical cell exactly like this. So most of our cells stay in G0, or a process where they don't divide. So like lung cells, neurons that are in the brain and our nervous system, they don't reproduce. They don't go through mitosis very often. Unlike cells like our skin cells, liver cells, they reproduce very readily. And so it's going to depend on the type of cell for how long G0 is. Some of them it's really a long time, some of them it's not very long. What comes after G0 is our G1 stage, and this is the first stage where the cell grows and becomes bigger because it just got reproduced out of mitosis. And so G1 is going to be used for the cell to reproduce organelles, start producing proteins, and a really important stage. The next stage of the cell cycle is going to be what we call the S phase. And the S phase is really important because this is where the DNA inside of the cell is reproduced. DNA is ×2. We do this with DNA replication, which I'll be talking about next. And then we have the G2 cycle. G2 is where we are preparing for cellular division, for the process of mitosis. And so there are 4 main stages of mitosis. We have prophase, and we have metaphase, anaphase, and telophase. Okay, and then this last step is called cytokinesis. And cytokinesis is actually where it's going to split. So these are this is called the M stage right here. So it's G1, S, G2, and M stands for mitosis, and these are the 4 steps of mitosis. At the end of cytokinesis, we should have genetically identical 2 daughter cells, and then these two will both go into a cell cycle of their own. Okay, so let's talk about these stages of mitosis a little bit more in depth. So prophase, if we were going to talk about from a chromosome standpoint, we're going to be looking at chromosomes that are going to be condensed into chromosomes. So during the G1 phase and the S phase, and G2, the DNA is just in the nucleus, just kind of all hanging out. And this is so cellular processes can take place, like protein synthesis and different things. When we enter into prophase, and to remember the steps of mitosis, it's PMAT. So prophase, metaphase, anaphase, telophase. So prophase is going to where in the chromosome we have condensing of DNA. And this is where it wraps around histones and makes little nucleosomes, and we get all of the chromosomes that we are going to have in our nucleus. During metaphase, all of these chromosomes and also the nucleus starts breaking down. During metaphase, the chromosomes meet in the middle. So metaphase, the chromosomes met in the middle. So if you were looking under a microscope, you would see this kind of darkened line in the middle, and that would be where the dense DNA would be lined up in the middle. We have little centrioles that are starting to like spit out some spindles that are going to pull these chromosomes apart from their centromeres. Centromeres is right in the middle, and that's where those spindles are going to be attracted to, and they're going to break apart. Next is anaphase, and anaphase, I always say Anna is a homewrecker, and so she's going to divide the chromosomes. So these spindle fibers that I was just speaking of are going to take half chromosomes right here. Okay, so these aren't arrows, but this is actually half of that chromosome being pulled apart. And then our last one is telophase. And telophase is going to be where we get two nucleus starting to form, and I always think of telescope, where, you know, something you look through, these kind of looks like binoculars. And now we have the DNA, the identical sets of DNA that we're going to get ready for cytokinesis, where the two cells separate and we get two identical cells. Cytokinesis is where the cytoplasm actually splits. Alright, it says, what is the structure of chromosomes and how does it form? Okay, so this is going to be referring to what I was talking about with the histones. Okay, so right here in the middle, this is called the centromere. At the ends, these are called the telomeres. And a half of a chromosome is referred to as sister chromatid. And so when we're talking about the chromosomes being split, we talk about these being split into sister chromatids, because sister chromatids are half of the chromosome. Okay, and then when we are dealing with the DNA being condensed, we start out with DNA, just the double helix DNA. It gets wrapped into histones, so this would be during prophase. Gets wrapped into histones, wrapped around histones. If you want to check this out, this is on page 131 of your book. There's a nice graphic there. And then those histones kind of start circling around each other, and so we get this coil, and then we get a supercoil, and then finally we end up with a very condensed chromosome that's full of just DNA and little histones. So histones, then we go into the first coil of just histones being wrapped around the DNA and starting to coil, and we call that chromatin. And from chromatin we go to supercoiled DNA, and then we arrive at the chromosome. So that's how it gets condensed. And then it says compare cytokinesis in plant and animal cells. Well, as you know, plants have cell walls, and because of this plate-like, cell wall that animal cells do not have, cytokinesis in an animal cell is kind of more like it's pulling apart. So in an animal cell, we have like this kind of cleavage that's formed between the two. So here's our DNA here, and so it kind of pinches. And in a plant cell, what happens is we have the two nucleus go on each side, and a plate drops in. So a pinches and a plate. So that's the difference between cytokinesis in plant cells and in animal cells. Okay. Now, next thing it says is to compare and contrast chromatin, chromosomes, and chromatid. So I just erased that picture, but if we were going to just review those, hopefully you would be able to tell me what those were. So chromatin, which one's first? Chromatin is going to be the histones with DNA on them being coiled. Okay, that it would be your chromatin. A chromosome is going to be a fully coiled DNA. Okay, and then a chromatid is going to be half of the chromosome that gets pulled apart during anaphase. Alright, the next part that we're going to talk about is DNA and protein synthesis. So now we're going to kind of zoom in on replication and what happens during these during the S phase. So the S phase, when during the cell cycle, when the DNA is being replicated, and then what's happening during G1 and G2 when the cell is carrying out its normal processes. Okay, so first, what is DNA? So DNA was discovered back in the 50s, and there was lots of scientists that kind of contributed to this discovery. And one of the principles that kind of allowed them to discover DNA was the fact that it could be transformed, or transformation could occur with DNA. So DNA is not a living thing. DNA is just a molecule. So just like it's a sugar molecule or something else, DNA is just a molecule that can be transformed into a different cell. So this is how we can do cloning, and this is how the early scientists like Griffith and some of these ones that we're going to talk about were able to find that DNA was actually a molecule that carried genetic information, and even if the cell died, DNA could still be transferred. So what is transformation of DNA? When DNA is moved from one cell to another. And we can do that because DNA is not a living thing. Okay, so the four nucleotides we have adenine that goes with thymine. And so we can just say that these are, we abbreviate them A and T. Okay, if we're going to look at their structure, thymine is going to be octagon that has some double bonded oxygens coming off. It's just a molecule. Okay, another one here. And adenine is going to be a stop sign and a house. That's what I kind of think of it as. And it as well has some different little molecules coming off of it. The other two are going to be cytosine, and cytosine is really similar to adenine. Or I mean, excuse me, cytosine is similar to thymine. It's just the stop sign, but it has some different structures coming off of it. And then guanine is similar to adenine, which is a G, and it is going to have a house and a stop sign all together. And A's always go with T's, and C's always goes with G's in DNA. Now we know later on when we start talking about RNA, RNA is different. Okay, so give a brief description of how the following people contributed to the discovery of DNA. Well, Frederick Griffith, he was the first one that did the transformation of DNA. And so Griffith's experiment, he did an experiment with different types of bacteria. He did S bacteria and R bacteria, and the S bacteria was a deadly bacteria and the R bacteria was not. And so he killed the bacteria, both of the bacteria, and then he transferred some of the bacteria from the S bacteria into the R bacteria, and even though the bacteria had died, they just moved the DNA over from the S to the R, and then it made the R bacteria also deadly. And so that showed that that DNA could be transferred between cells, and this was a really big discovery for this time period. Rosalind Franklin, she was the one that took the picture of with an X-ray. So she took an X-ray picture of DNA, and this helped Watson and Crick, who's next on our list. They were credited with the discovery of DNA in 1953. Watson and Crick. Oswald Avery, he identifies that DNA in fact does have a transforming principle, which he basically built off of this experiment of Griffith, and he really wanted to prove that that was really what was taking place. And so he did a series of tests. He did chemical analysis, he did enzymes test, and his group finally had evidence to support that DNA was genetic material, and so that was being passed down from generation to generation. And then Hershey and Chase were kind of the last ones that made a contribution, and they used bacteriophage. And bacteriophage is actually, it has the word bacteria in it, but it's actually a virus. And it's the little robot-looking man that looks like this, that attaches to a cell and drops its DNA in there. And they were able to see that bacteria infected with the bacteriophage would receive the DNA from that virus. And so they were able to conclude that the virus DNA was able to be passed down or passed into the cell, and the protein was not being passed down, so that gave them the conclusion that DNA was being passed down was not a protein, and that the protein wasn't holding the genetic material. Okay, so we already kind of talked about the structure of the nucleotide, so we want to talk about DNA replication now. So DNA replication, we get to talk about enzymes again, and those, remember, are proteins in the body that allow chemical reactions to take place. And this is the first one that we really investigated this term. So enzymes in DNA replication consist of DNA polymerase, and helicase. Alright, so we have a piece of DNA, and so we have our A's and our T's and our C's and our G's matching together. And what happens is DNA polymerase, or helicase, comes in right here, and it attaches to the DNA, and it's a protein, so it's just kind of this structure that sits right there, and it spins as fast as a jet engine, and it unwinds the DNA, and so it's able to uncoil the DNA to expose these nucleotides, so that we can replicate DNA. And so this is happening during the S phase of the cell cycle, and mitosis would not be able to occur without DNA being replicated. So we have two DNA polymerase, one helicase. Helicase is breaking it apart. There's a little replication fork right here that's allowing this. And now the interesting part with DNA polymerase, one moves from the back direction from 5 to 3 prime, and the other one moves forward. So we would have a DNA polymerase right here, and we would have a DNA polymerase right here, and this one would be moving in this direction. It kind of loops around and continues to replicate, and this one moves this way. So as the DNA polymerase is moving, it actually is gathering nucleotides from the nucleus, which I'll be able to explain where those come from later, and it's finding the right match. So if this was A right here, the DNA polymerase finds a T, and it not only finds nucleotides, but it also proofreads the nucleotides as it was being produced. So this piece of DNA is going to stretch out along here, and then this is doing the same thing. It's building a piece of DNA from this way. And so what we get when we are done is we have only a semi-conservative piece, two pieces of semi-conservative DNA. So what that means is if we conserve something, we're making it from the old. And so we have an old piece of DNA here and here, and we're making two new pieces of DNA, so it's only semi-conserved. Okay, so the result of DNA replication is 2 new strands of DNA. Okay, and they're exact copies, and so these are going to form those chromosomes in prophase, and then each of those daughter cells at the end of mitosis will both have one full strand of DNA. Okay, so one will have this one and one will have this one that's being formed. Okay, this again happens in the S phase. Okay, now so transcription translation. So this is what's happening the rest of the time. Okay, so this is what happens in the S phase during G1. What is happening is the regular processes which of the cell, which is to produce proteins. Okay, so we're inside the cell, inside the nucleus. The DNA is just strung out. It's not condensed into its chromosomes, and transcription is going to be for one gene. Okay, so I think humans have around 20 or 30,000 genes, and transcription is for one gene, because one gene is going to equal one protein. Okay, so one protein is the result of this whole transcription translation process. Okay, so transcription. We have one gene of DNA, okay, from beginning to end. We're going to have markers on those genes. Transcription factors are what they're called. So we have RNA polymerase, we have the Tata box, and we have a whole bunch of different proteins that all assemble at that promoter and region there. We have RNA polymerase, which I already said that one. We also have the enhancer, which is a little further down, and it's going to come into this region of the Tata box. This piece of DNA is going to bend around. Okay, so if it was to move up here, so it's going to look like this, and that enhancer is going to touch back on itself. And when that happens, when this enhancer, the same one that was right there, connects here, that allows a chemical reaction to take place. It's going to allow RNA polymerase, which is a lot like DNA polymerase, to start copying this one gene. Okay, and so this one gene might be for, eye color. It might be for, hair color. It might be, the many different genes that we have in our body. It's going to copy this one piece of DNA. Okay, so what does RNA polymerase do? So, similar to what DNA polymerase does, we have two strands of DNA here, well, one strand, two-sided, come in, and what happens is there is a little place that is going to separate out the DNA, and in that amount of time it's going to build new DNA into, or new nucleotides. Okay, and so what we get is we start getting this long chain of single-stranded RNA. So RNA makes RNA polymerase. Okay, and then instead of leaving it open, the RNA polymerase actually closes the DNA back up and returns it to its normal state. So what we have is just this long piece of RNA that is produced, and when the RNA polymerase gets to the end of the gene, it'll stop, and this piece of RNA will then leave. And it heads over to what is called editing. So editing is a very important step in transcription, and transcription is going to be finished once that RNA is produced. So we're going to have this long strip of RNA, and editing is going to take place, and it's going to cut out the exons and the introns. So exons are going to exit, and that means that they are going to be, whoops, I didn't mean to erase that. The ones that are going to head out and be part of translation. So if this is an exon, this is an intron, this is an exon, and these are just regions of DNA. Now the introns are going to stay inside of the nucleus, And these are going to go back up, and right here where these new nucleotides are being found, right here, these little guys are coming in at a very fast rate. And so there might be A's and T's, like this might be an A, this might be a U, C, a G, okay, because RNA, there's no T's. And those come from the introns when it's cut out. So what we're left with is just the exons, so they're all exons. It's going to shorten this, and this is going to be called our mRNA, which the M stands for messenger, and it's going to exit the nucleus. Okay, so after it exits the nucleus, we are going to meet up with the second type of RNA. So our first type of RNA is our mRNA. It's going to leave the nucleus. Our second type of RNA is rRNA, and that is ribosomal RNA. And so it is actually nucleotides that go through and form these two subunits. And our RNA is a ribosome. And this is where proteins are made. And then our last thing that is part of it is the tRNA. And tRNA kind of are made up of DNA just the same way. They're going to have three little nucleotides that we're going to call the anticodon right here. So this might be an A and a C and a G. Okay, this is our anticodon. The mRNA that lands right here, each three of these are going to be called our codon, and they're going to be attracted to each other. So what happens is the tRNA comes in and it binds with the codon if it's the right code, so it has to be an A matching with a U, a C matching with a G, and a G matching with a C, and a U matching with an A. So mRNA, our nucleotides are A's with U's and C's with G's. And this is really important because what it's carrying is a amino acid. And all of these have a different, there's 23 amino acids, all of them have a different one. And as these things come in and join, these tRNAs come in, they're going to start building this polypeptide chain of amino acids that eventually is going to fold into a protein. So our three types of RNA are going to be mRNA, rRNA, and tRNA. Protein is made at the ribosome through anticodon and codon matching up and the amino acids making a polypeptide that turns into a protein. These proteins could be used to make your heart beat. They could be made to make the skin color the way it's supposed to be. Whatever that gene, that one gene, was being coded for is going to be produced in that one protein. Okay? Now we have some different mutations that can occur. Okay, and so there are mutations that can happen because if there's a mutation in the DNA at this point down here that then is copied into RNA that then goes to the ribosome and starts, making this protein, and there's a problem with one of the nucleotides, that's going to cause major problems for whatever that protein function. And sometimes proteins don't function at all. Sometimes proteins have the wrong function. And so mutations are caused by problems with the DNA that then get made into proteins that then have the wrong function. So there are gene mutations and there are chromosome mutations. And gene mutations are going to be ones that just deal with nucleotides. Okay, so maybe we have like an A and a T and a C, and a G and a G and an A, and it's supposed to have a C here, but it gets deleted. Okay, so deletion is 1. And so that scoots everything over 1 this way. Or there could be something inserted, which would be, so you have A, T, C, G, G, A. So if our regular one was A, T, C, G, G, A, C, C. Now let's not make that a C.
Good afternoon.
Okay, so if that was what it's supposed to be, then this one was deleted, and let's say there's an A and a T here, then this becomes, it's deleted, and it's a double A and T, and then everything past this has been moved this way, whereas this one, if it's there and we get another letter there, now all of a sudden everything has been moved that way. So all the proteins that are produced past this are going to be incorrect. All the proteins that are produced here. And we call these frame shifts. And this is a big problem because, as you know, like if we have a piece of mRNA and it's A, U, G, G, A, C, T, oop, no T's. Just kidding. CG, and we code that. So if these are each of our codons for translation, our tRNA is going to come in, and we're going to have a U, an A, and a C. It's going to be the opposite. C, U, G, G, C, G. So these are anticodons, and there's going to be amino acids for these. And this is where we're going to use the codon wheel. And the codon wheel is, so to use this wheel here, if our first nucleotide is a U, then our second one is an A, and our second one is a C. It's going to be tyrosine. Whoops. Tyrosine. This one would be, first nucleotide is a C, second one is a U, third one is a G. This would be leucine. This one, first one G, second one C, third one G. This is going to be alanine, A-L-A. Okay, so then these are the amino acids that are eventually going to build our protein. So if everything is shifted over 1, all of these amino acids are going to be different than the ones they're supposed to, which is going to create a different protein, which is going to not function. So it's a pretty big problem. Alright, let me get rid of this. Okay, and then the next thing we have are chromosomal mutations. And chromosomal mutations are a little different. That's after the chromosome is already done, and this happens mostly in meiosis, but we have a chromosome, and it can be 4 different chromosomal mutations. We can have a gene that actually gets duplicated here, a section of a chromosome. So maybe we have this green part gets duplicated. So when we have it over here, we have 2 sections of this. You can also have, so we have duplicated, we also have something that could be deleted. So then maybe this green section is deleted, so then the top of your chromosome is shorter because that section is missing. You could also have what is called an inversion, and so this whole section gets flip-flopped and turned upside down. And I think on the test we had it like as A, B, C, D, E, F, G, H, I, J, K. And so if this was the different genes of a chromosome, this part could be deleted. So we'd have A, B, C, G, H, I, J, K. So we'd see that was a deletion. If we had something that was duplicated, then our answer would be like A, B, C, D, E, F, D, E, F, G, H, I, J, K. If something was inverted, it'd be like A, B, C, and then F, E, D. So this is flip-flopped here. So that's the inversion. So deleted, duplicated, inversion, and then the third one, translocation, might be like ABC, and then we go GHI, and then the DEF comes in, and then we have JK at the end. So this part was translocated from this place, or from here over to there. All right, let's see where we are. So there's the mutations. Chromosome mutations. Okay, so now we're ready for meiosis and genetics. Okay, so meiosis, remember, is what happens to make our sex cells. Okay, so we have autosomes, or somatic cells, and we have sex cells. And sex cells use meiosis, and somatic cells use mitosis. So what we've been talking about so far, somatic cells, that's everything we've been talking about so far. Now our sex cells, which are sperm and egg, go through the process of meiosis. And it's a process, not a cycle. Okay, so it says, what's the difference between autosomes and sex chromosomes? So autosomes are going to be what if we have our karyotype here, and we have our 23 pairs of chromosomes. All of these are going to be autosomes, except for, so all these are chromosomes, okay, except for 2 of them. And these 2 chromosomes are either going to be XX or XY. XX is going to be the girl, and XY is going to be the boy. So sex chromosomes are going to be what determines the girl or boy. And these are also the 2 types of sperm that are produced. We have XX sperm, and we have X sperm, and we have Y sperm, and eggs are always X's. And so that's how we get boy or girl. So if the X sperm binds with the egg, your baby's going to be a girl. If the Y sperm binds with the egg, it's going to be a boy. Okay, and so gametes, they're going to be the sperm and the egg. They are formed through the process of meiosis. And it's going to basically be the process of mitosis. We're going to do prophase, and then we're going to do metaphase. And what we're starting with here is called a germ cell. And germ cells are both what females and males have. And these are in the ovaries and in the testes. And these germ cells go through the process of meiosis to produce gametes. Okay, so prophase, same thing's going to happen. We're going to have all of our chromosomes are condensed, except for now in a germ cell, we actually have double the amount of DNA. We actually have our mom and our dad's DNA in our germ cells. So we have double the amount of DNA, and through these 2 processes of, or these 2 rounds of dividing, they're going to increase genetic variability by the process of crossing over, which happens during prophase 1. So we have crossing over, and what crossing over is where some of these chromosomal mutations can actually take place. So we have a chromosome from our mom, and we have a chromosome for our dad, and they lay real close to each other, on top of each other in the nucleus. And what happens is certain parts of them switch. So maybe this part turns red right here, and then this part of this chromosome turns green right here. So they switched these 2 areas of the chromosomes, so that increases our genetic variability there. And these chromosomes are called homologous chromosomes because they're the same. And we're going to mix them up a little bit so that we have genes from both our mom and our dad. So these are not the same DNA that is in our cells that make us us. This is our parents' DNA. We have 46 pairs, 92 chromosomes total in here, so it's a lot. Germ cells have a lot more DNA than a regular somatic cell. Okay, metaphase 1. This is chromosomes meet in the middle and they're going to divide. So this homologous chromosome is going to go on one side into one cell. This homologous chromosome is going to go on the other. So anaphase 1, this is where the chromosomes actually split. And then telophase is our first division where we get, telophase 1 is where we get our first 2 new cells form, and each have one of the homologous chromosomes. So if we were going to depict that and with just one chromosome, we would have this one, a cell, and we have this one. This one would have this bit here. Okay, and so our chromosomes had this little bit of variation, and there would be 46 more, or 45 more in this one, 45 more in this one. So it's 2 full sets. So these are called 2 diploid cells. All right, so now we're going to split these cells again. And to do that, we're going to go through meiosis 2. So what happens here? We go through prophase 2. And prophase 2 is going to be, they get ready to divide. The centrioles are going to spread out to each side, and we are going to get ready to divide again. So prophase 1 is not as nearly as important as prophase 2 because there's no crossing over that takes place. So just gets ready to divide. And then metaphase 2, now our chromosomes are actually going to split in half. So we have our 2 chromosomes here. Okay. And now these are actually going to divide in half. So this is in one cell, this is in another cell. So now these are going to divide in half. So the spindles are going to lock up at the centromeres. And during anaphase, the chromosomes are going to split into sister chromatids. Divide, and so we actually just have these like little half chromosomes in each of the 4 cells. So now we have something that looks like this. Of course, this little bit is green. And then we have another one that is all red. We have one that is only a little red here. Oops, that's gray. And then we have one that is all green. And this is just, remember, one of the chromosomes in the nucleus. Now I'm just doing the half. Okay? And then in telophase, we get 4 haploid cells. Each of them have 46 half chromatids, or you could say each of them have 23 sister chromatids. So either way that it makes sense to you. And now these are going to turn in to our sperm and egg. So if it is a sperm, each of these is going to, in the lining of the testicles, this is going to turn into a sperm. This is going to turn into a sperm. This is going to turn into a sperm. And this is going to turn into a sperm. If it is an egg, we're going to create some polar bodies, and we're only going to get 1 egg here. So we're going to get 1 egg, and these 3 are going to be these are going to be polar bodies that don't form anything, that just get reabsorbed, and the DNA gets those molecules get used elsewhere. Okay, So haploid, these are the haploid cells, and haploid just means half, okay, and half of the DNA. Diploid cells are going to be the first cells that we created, these ones right here, that have the full DNA. Okay, process of meiosis 1 and 2. How is it different than mitosis? Well, there's some crossing over that takes place in prophase 1. But the big thing is A, it's not a cycle. This cell isn't going to go through meiosis again. These are now gamete cells, sperm and egg. And in mitosis, the daughter cells go back into mitosis and they're going to do the same thing over again. In meiosis, that doesn't happen. So then, of course, our sperm and egg then will have the ability to go through fertilization. And fertilization happens during sexual reproduction, so the sperm and egg will unite, and then once those two come together, we will have a blastocyte that is formed. So we first have meiosis that takes place. We form our gamete cells, which are the sperm and the egg. And then those two come together through the process of fertilization. And that's going to make our baby, or not necessarily a baby, I guess. We have the blastocyte. And then a blastocyte can develop into a baby, or these are our totipotent stem cells that can be used for all sorts of curing of diseases and genetic problems. Because stem cells can turn into any type of cells. They're just unspecified cells, undifferentiated cells. Alright, so now going into Mendel's experiment. Mendel used pea plants to look at this genetic DNA being passed down into its offspring. So pea plants, they have, they reproduce pretty quickly, and he was able to control what they mated with. And so what he did was he took two of the a purple flower, which was big P, and he took a white flower, which was little p, and he crossed them. And what he got was, he got 4 flowers, and they were all purple. And at that time, they thought blending was something that took place. And since they were all purple, and this one was white, that was kind of thought that, well, maybe the white just was overdone by the purple. Well, he went on he did one more cross with these. These were our homozygous, dominant, and recessive. So this one would be our dominant one, and this one was recessive. And what he made in his F1 generation was heterozygous, dominant. So they were all still purple because they were dominant because of them having this big P. But when he crossed them again, what he got was, he got big P, little p, big P, little p, big P, little p, and he got one little p, which turned out to be white, and not a blended white, but a full white. And we got a homozygous recessive, which showed that these recessive genes were being passed down. And just because they were all purple didn't mean that they didn't carry the ability to become white. And so this was how our law of segregation and was formed by the crossing of these. And he used the Punnett square. Oops. He used the Punnett square to do these probabilities. So if we were going to do that one that we just did, I could show you a monohybrid. Monohybrid is going to be one trait. The ratio is just going to be like 4 to 1 or 3 to 3. So we have big P, and then we had little p, little p. So everything is going to be like this. Okay. But if you crossed a big P with a little p on monohybrid, you'd get big P, which would still be purple, big P, which would still be purple. Then you would get a 50% chance of having white. So this would be, you know, the odds of getting one would be 2 of 2, where in this one, it'd be 4-0. Okay, so square by understanding homozygous, heterozygous, dominant, and recessive. Okay, we talked about those. All right, now this is going to be, this right here is an allele. Okay, and an allele has has 2 genes. So we have gene 1 and we have gene 2. Now, in this case, gene 2 is not going to be expressed because gene 1 is dominant. And so this is going to be your genotype. Okay, your phenotype is going to be what it looks like. So if this is our genotype, then our phenotype would say that this is going to be purple. Okay, our phenotype for this one would be white. So phenotype is just going to be what it looks like. Okay, so this is a monohybrid. A dihybrid, on the other hand, is going to be for 2 separate traits. And so if we were to take maybe eye color and hair color, so we had brown hair and brown eyes, okay, and we crossed it with somebody that had blonde hair and blue eyes, now we have 2 traits that we need to do. So I know some of the teachers did like kind of a foiling method, but I like to do the 2 monohybrid crosses. So I do a monohybrid for hair color and a monohybrid for eye color. So I would take my B's and I put them in the first one. So I have big B, little b, little b, little b. So I have big B, little b, little b, little b, big B, little b, little b, little b. And those I'm going to put across my dihybrid top. So I'm going to have big B, little b, big B, little b, little b, little b, little b, little b. And so I just took that one and put it here, that one and put it here, this one there, this one there. Okay. Then I'm going to do the same thing for eye color. So big E, little e, little e. So I got those from right there. Big B, E, little e, big E, little e, big E, little e, big E, little e. So those are all the same. And now the dihybrid cross is going to go inside of here. Okay. And so you can see none of these are going to have blue eyes. They're all going to have brown eyes. And then we're going to have some of them that have brown eyes and blonde hair as an option, or we're going to have brown eyes and brown hair. And so from this, when you count up how many they're going to be, you can get like a 4 to 4 to 4 to 4, because it explains like both of them are dominant here. First one's dominant, second one's recessive. First one's recessive, second one's dominant, and then this one says both are recessive. Okay, so then we have 4 exceptions.
Anya?
So there are 4 exceptions to these laws that Mendel put into place. And basically what they are saying is that there's not something that's dominant and something recessive always. Sometimes we can have 2 that are dominant and they both are expressed, or we can have 2 that are dominant and 2 that are recessive that are all being expressed. So those 4 are going to be something that's called incomplete dominance, which is going to be a blending of 2 dominant traits. and we see this in the betta fish, and also with red and pink flowers, 4 o'clock flowers. We have codominance, and codominance is going to be both are dominantly expressed, except for they're going to be like spotted. So codominance would have like a black and white, a black mom and a white dad chicken that reproduce, and the chicken comes out spotted. Or blood type. Blood type is also like this. And we could, I've done a Punnett square in a different video that explains how that is codominant. Then we have polygenetic traits, and this is like your eyes. And your eyes can have multiple dominant and multiple recessive. That's why poly, multiple. And they can actually all be expressed, and so that's why eye colors vary so much. And then our last exception is epistasis, and this is the blocker gene that blocks all other genes, recessive or dominant, and this is the reason that we have albino traits. Okay. Crossing over, you know that we kind of already discussed that one. Alright, just a little bit left here. I'm not going to go as in-depth with these just because these aren't as far away. We just did these in class not too long ago. Okay, so Gregor Mendel is considered the father of genetics. So who would be the father of evolution? Well, that would be Darwin. Darwin discovered natural selection at the Galapagos Islands, and he saw that animals had adapted to their environment, and kind of started this whole theory of evolution. Evolution is just a change over time. Change over time doesn't mean that something has evolved from something else. It's just over time natural selection has occurred, and it has changed an organism. Okay, natural selection is when the environment changes, and when that environment changes, the species can has two choices. It either dies because it doesn't have what it takes to survive, or it has some kind of special trait that allows it to survive. And then if this one dies, that trait is going to die out, and if it survives, then this trait is going to be passed on, and that's how we get to adaptation. So these traits allow a species to adapt to an environment. But this isn't going to be in one lifetime. This is going to be over many generations. Okay, analogous structures, homologous, and vestigial structures. So analogous is going to be different organisms that have a similar trait, but they're not related. So this might be the wing of a fly and the wing of a bird. Okay, these two species are not related, but they have similar, they both are able to fly with wings. Where homologous structures or homologous structures are going to be structures that are the same, except for they're in different species. So like the pelvis of the whale that was found, and the pelvis that we have as humans. I don't know how my pelvis drawing looks, but not good. And then the last one is the vestigial structures. These are going to be structures that were maybe used in our ancestors, but we don't need them now. So like the appendix. For humans, we don't use our appendix anymore, or maybe wisdom teeth. Okay, what is artificial selection? Artificial selection is going to be humans. Humans breeding animals to get traits that they want. So I know some of the teachers showed the super cow, and the super cow is this really beefed up cow, and over generations they've only bred, you know, the most muscular bulls with the most muscular heifers, and over time we've gotten a super cow. Or like breeds of dogs, we've bred the runt of the litter over and over to get the little teacup poodle, or we've done, you know, other breeding to get really big dogs. And what's the main difference between artificial and natural selection? Humans. One is done by nature, hence the word natural, and the other one, artificial, is done by humans. Alright, this is just what we talked about this week, the levels of organization, population of a species. So population is going to be the same species that is in population. Is what ecologists look for when we're looking for changes in environment. 5 types of consumers. Okay, so we're going to have, okay, so we have our 2 main classes. We have autotrophs and we have our heterotrophs. And autotrophs are going to be our producers that are going to make their own food by two main ways. They're either going to go through photosynthesis or chemosynthesis, and chemosynthesis is going to be our bacteria. Okay, heterotrophs, these are going to be our consumers. And we have 5 of them. Our first one is going to be our herbivores. And herbivores are going to eat our plants, our producers. Our second level is going to be our carnivores. And our carnivores are going to eat meat. Then there is our omnivores. And omnivores are going to eat both plant and meat. And then we have our detritivores, and they are going to be eating dead matter. And then our last one are our decomposers. Oh, and the detritivores are also going to eat other meat and like a carnivore would. And then our decomposers only are decomposing all of our organic matter. So getting all of the organic molecules, so carbon, hydrogen, oxygen, phosphorus, sulfur, back into those atoms so that they can be used by our plants again to keep the cycles going, which we learned about in our biochemical cycles that I will talk about here in just a second. Okay, and then food chain versus food web. So food web, we, you know, if you have a, you know, plant that's going to put its energy into a rabbit that is then going to put its energy into a hawk, and then the hawk then puts its energy into a fox, so the fox eats the hawk, hawk eats the rabbit, rabbit eats the plant. We might have many different food chains in an ecosystem, but that doesn't really give us a realistic picture of what's going on in that ecosystem. So what we really need is multiple food chains showing us all of the different avenues in which something can transfer its energy. And then when we construct all of these together, then that would show us a real food web. So then we might have just a plant here, and from the plant could go rabbit, or it could go to the cricket. And then we start getting a more complex Then the cricket could go to the rabbit, and then the rabbit goes over to the fox, and the rabbit could also be eaten by the hawk, and then the hawk can also be eaten by the fox. So we get more of a complex, maybe a more complex look at what an ecosystem's food web really looks like. Okay. So biodiversity is going to be when we have lots of different organisms. Obviously, biodiverse regions like rainforests are going to have lots of different species of life compared to maybe a desert when there's, just a few species because of lack of resources. So closer to the equator that you get, the more biodiversity you have. Further away from the equator, typically the less biodiversity you have. Okay, so detritivores and decomposers are different because detritivores are going to eat our dead things, and it's kind of a second step to our decomposers, where our decomposers aren't really eating anything. They break down things by using that chemosynthesis. Okay, explaining the trophic levels. Okay, so once we have these levels over here, we can start talking about the amount of energy that is going to be flowing. Okay, so we can show this in a pyramid. Okay, so if we have a pyramid here, these are going to be our producers at the bottom, getting their energy from the sunlight. Okay, and then we're going to have our first level consumers. So this is obviously our herbivores. Then we're going to have our second level, which is going to be our second consumers, and then we might have third, and then we could even have fourth, and we're always going to be up here. This is us, we're at the top. But notice, if you knock off the top of the pyramid, everything else stays intact. If you take out the bottom, all of these things will die. So when you're thinking about the energy that it moves upward, nobody needs our energy, so we're really not needed in an ecosystem in comparison, especially to our bottom level producers. Okay, we want to talk about the biochemical cycles real quick. And so our biochemical cycles are very important for all life on Earth. If any of these atoms were to go out of balance, or any of these cycles, plants would cease to exist, and if we didn't have plants, we wouldn't have oxygen. If we didn't have oxygen, we wouldn't be here. Also, plants serve as that bottom line producer in our energy pyramid there, and so you could see that if any of these cycles were not functioning correctly, then we would have no life. So, carbon cycle, what organisms contain carbon? Well, all organisms contain carbon. The process involved in the carbon cycle is going to be with CO2, carbon dioxide being produced by automobiles, factories, by humans, by animals, and carbon dioxide is needed by plants. Okay, so obviously there's a lot more of these than those, so the carbon dioxide cycle isn't going quite as equally as we would like it right now, and this is some of the factors that are contributing to global warming. The nitrogen cycle, nitrogen comes from the atmosphere. We have bacteria that is nitrogen fixating. This is the process of chemosynthesis, so they turn it into ammonia. Then we have more bacteria that turn it into NH4, that then have more bacteria that turns it back into N2. So this is a really important cycle for bacteria and also for us, because this NH3 is needed for plants to grow. And without ammonia, our plants wouldn't grow, and again, we wouldn't exist. And then our last cycle that is pretty important is our phosphorus cycle. And phosphorus comes from rocks and runoff from when it rains. And so we need phosphorus in our diet. We get it from the plants. So we have rain that comes down, leaks off of the rocks, and phosphorus goes into or phosphates go into the soil. The big thing here is phosphorus never goes back up into the atmosphere. It stays in the ground and in the soil, and it's used by plants, and then we eat the plants, and then when we die, the phosphorus goes back into the ground, and it's again used by plants again. So it is a cycle, however it does not go into the atmosphere. Alright, I hope this helped you study for your final, and have a great evening.